Hello, my name is Albertura from University of Oviedo in the north of Spain. I want to present you our work about novel tools for early detection of a global aquatic invasive, the zebra mussel, Dreissena polymorpha. One of the current widely recognized anthropogenic threats to marine biodiversity is the cross-regional transfer of non-indigenous species causing irreversible alteration in communities, habitats, and ecosystem functioning. Due to the need to stop the spreading, have developed different strategies: prevention, for keeping out invasive species; early detection and rapid response, for detecting and eradicating invasive species to stop them from spreading; control and management, applying integrated control techniques to manage the problems; and rehabilitation and restoration, heal, minimize, or reverse the harmful effects from invasive species. Early detection of non-indigenous species, and particularly potentially invasive species, is a prerequisite for the efficient and timely response. Further, spread prevention, eradication, if it's possible, and mitigation of threats to ecosystems and economy. However, at the early stage of incursion, the species is likely to be sparsely distributed in the ecosystem, reducing the chances of detentions by routine surveillance methods. On the other hand, the dispersal life stages, such as planktonic eggs or larvae that might be relatively abundant, are not identified in traditional zooplankton surveys owing to their cryptic morphology and lack of sufficient species-specific taxonomical expertise. Among many registered nuisance organisms, the zebra mussel Dreissena polymorpha is recognized as one of the worst invasive species worldwide. For that, the present study is focused on the development and validation of species-specific molecular markers for detection of the zebra mussel from environmental samples, in this case from plankton samples. For example, in 30 years, since zebra mussel was introduced in North America, it has become one of the world's widespread and abundant freshwater invertebrates, and has fundamentally transformed freshwater, food webs, and biogeochemistry. For tuning up the genetic tool, together with Dreissena polymorpha, another 5 bivalve mollusk species with a planktonic larval stage were sampled from the Lithuanian coast on the Baltic Sea: the native Cerastoderma glaucum, Macoma baltica, Mytilus trossulus, the already established invader Rangia cuneata, and the cryptogenic miarnaria. Cross-amplification was negative in all cases. At the next validation stage, the sensitivity and specificity of the designer primers were experimentally verified. Successive dilution 1, 1, 5, 110, 150, and 100 of the Dreissena DNA in distilled water were used as template for PCRs. The method allows to detecting at least 0.7 nanograms per milliliter of Dreissena DNA. Agarose gel and capillary electrophoresis were used for estimating the applicant size and additional validation tests of all PCR products obtained in the course of this study. For field validation of the designed marker plankton samples from the location where Dreissena polymorpha could be potentially detected were used. NGS metabarcoding, using cytochrome oxidase gene CO1 as DNA barcode, was employed as an independent method for confirming the presence of the Dreissena polymorpha DNA in the environmental samples. Phylogenetic information helped us to detect the region with the less intraspecific and most interspecific variation for the design of species-specific primers. This study presents a newly designed robust species-specific marker that search for detecting the presence of the global invasive species Dreissena polymorpha from environmental samples, even at very low concentration of the species DNA. The marker can be fully genotyped in a few hours, requires only minimal technical knowledge, especially if it is visualized in agarose gel. The molecular tools presented here are technically accessible and will facilitate the monitoring of zebra mussel population at a minimal cost.